Okay. Hello to everybody who's watching. And also special greetings to Buretno, our lecturer in organic chemistry. So this is our group presentation for when we are Group C, and then my name is Jason Alexander Yusuf. I'm with Cantika Diva, Bianca Martawardaya, and Arden Fadhilla. Next. And so here are a few of our key points that we will discuss, which are amino acid structure, amino acid synthesis, reactions, peptides, as well as peptide bonds. Next.
All right, so let's start first with amino acids and their general structure. What is an amino acid exactly? Amino acids are constituents of proteins. That is, when multiple amino acids combine together, they're going to form proteins and they help our body functions. As per their name, right, we have an amino, which is an amine group, and an acid, which is in this case a carboxyl group. Naturally occurring amino acids have the amino acids that are bonded to the alpha carbon of the carboxyl group, and they're thus called the alpha amino acids. And if you can see here on our figure on the left, we have the amine group, the NH2, that is bonded with the alpha carbon in the carboxyl group, with an R group attached that becomes the identifier. And if they are to combine together through polymerization, they create polyamides and they form proteins, which we use in our bodily functions. Next slide, please. Okay, so naturally occurring amino acids, right? We have 20 that we have identified, and we have 10 that we produce within our bodies itself. The identifiers here that distinguish all these amino acids are the R groups, or the side chains. And the constituents of these side chains are the ones that's going to become the identifier of these amino acids. Now, we also have amino acids that exist in a chiral center, right? So we're going to have two enantiomers, which is called a D and an L amino acid. There's only the L amino acid that we naturally use and naturally exists. So it's the only one that is naturally useful for us. And out of all the amino acids, some may have some alkyl groups, some may have some additional functional groups as well. And then quite interestingly as well, most, if not the majority of amino acids are primary amino acids or primary amides, right? Aside from one, which is a proline, which is a second degree amine, and also a secondary amine, and also two amino acids which are called isoleucine and threonine, which are also another stereocentric, which has a different form from most other amino acids. Next slide, please. Okay, so let's talk about the acid-base properties of amino acids. So we can tell from the name, right? We have an amine group, we have a carboxyl group. The carboxyl group has a tendency to be an acid, while the amine group has a tendency to be basic. So we also, quite interestingly, we have these naturally occurring forms of amino acids that is called zwitterions, which only exist at the pH of around 6. While the neutral amino acids here can have an overall charge depending on the pH, right? And the zwitterion here is the neutral form of this amino acid. Yes. Next slide, please. Okay, so next, let's talk about the amino acid synthesis. We're gonna talk about 3 methods of synthesis. First is the SN2 reaction with alpha halo acids and NH3. So alpha halo acids, right, have the X, which is the halogen that's bonded to the carboxylic acid group. And if we are to react this with an NH3 in excess, we're going to have an amino acid. And here we're going to have an SN2 reaction mechanism, which we have learned about before, with the halide as our leaving group and the NH3, which acts as a nucleophile. Okay, next slide, please. Okay, second is the alkylation of diethyl malonate derivatives. So this is the based on the malonic ester synthesis from diethyl malonate to create diethyl acetyl metamalonate, which we can react with an AOH, and we're going to have the following mechanism with the deprotonation, the alkylation, and the hydrolysis and decarboxylation. to finally form the amino acid that we desire. Next slide, please. And last is what we call the Strecker synthesis, which primarily uses ammonia, cyanide, and an aldehyde as precursors to the functional groups of amino acids. So here we have the following equilibria involved, right? We have first the aldehyde reacting with NH3, and then we're going to have it react with the cyanide, sodium cyanide, and we're going to react with an acid to finally find our amino acids in either L or D form. Okay, next slide, please. Okay, now to get the final version of the amino acid that we need, right? As we have heard before, the synthesis of amino acids will create either L or D amino acid, in which only the L amino acid is actually useful for us. And so the synthesis also is also followed by a separation method, in which there are two. One is through the diastereomer method, and the second is through using a chiral reagent. And both of them takes advantage of the different physical and chemical properties of the stereoisomer that's reacted with another chiral reagent, so that we can actually separate them and get the isomer of amino acid that we can actually use for our bodies. Next slide, please.
The next is reaction due to amino acid. First is reaction due to amino group. Oxidative deamination. An amino group is removed and corresponding alpha keto acid is formed. Alpha keto acid produced is either converted to glucose or ketone bodies or is completely oxidized. So as you can see on the picture, first is amino acid and then remove 2 atoms of hydrogen becoming amino acid, and then put water into the equation and become keto acid. Next slide, please. Second is transpination. So transpination is transfer of an alpha amino group from an amino acid to an alpha keto acid to form a new amino acid and a corresponding keto acid. So the question is just like picture below. Next slide. Third is formation of carbamino compound. So CO2 binds to alpha amino acid on the globin chain of hemoglobin to form carbamino hemoglobin. The reaction takes place at alkaline pH and serves as a mechanism for the transfer of carbon dioxide from the tissues to the lungs by hemoglobin. As you can see on the picture, it's the carbamino group 4. Next slide, please. So next is reaction due to carboxyl group. First is decarboxylation. Amino acid undergo alpha decarboxylation to form corresponding amines. For example, is glutamic acid become GABA, histidine become histamine, and tyrosine become tyramine. As you can see on the picture, this is the reaction of histidine becoming histamine. Next slide, please. Second is formation of amide linkage. Non-alpha carboxyl group of an acidic amino acid reacts with ammonia by condensation reaction to form corresponding amides. For example, is aspartic acid become aspargine, glutamic acid become glutamine. And from this question below, this is the example of aspartic acid becoming an aspargine. Next slide, please. And then reaction due to both amino and carboxyl group. Formation of peptide bond. So carboxyl group of an amino acid binds with amino group of another amino acid, forming a peptide bond with the loss of one molecule of water. As you can see on the picture, this the equation for peptide bond, and then the hydration reaction occur, removing water, and then we can see the peptide group form after the reaction. Next slide, please.
Okay, so next I will be discussing about peptides. This is actually mentioned briefly by Vivi and also Fadil about peptides and peptide bonds. So what actually is a peptide? Peptides are actually from the Greek word, which means digested. They are short polymers of amino acids linked by peptide bonds. The covalent chemical bonds form between two molecules when the carboxyl group of one molecule reacts with the amino group of another molecule. On your right, you can see a simple figure of how it actually is. A peptide is a chain of it, a combination of a lot of amino acids. And on the right side, you will see the C terminus, which is the rightest part of the carboxyl group, and the left side, which is the N terminus or the left side of the amino group. Okay, and next is what actually differentiates an amino acid, a peptide, and a protein. So this is actually, again, mentioned briefly by Bibi before. I'm just going to review it a little bit. So amino acids are small compounds containing an amino group and a carboxyl group. And it contains a side chain structure that varies between different amino acids. So you differentiate one amino acid from another based on their side chains. And meanwhile, a peptide are chains of amino acids that have linked together by peptide or amide bonds, which will be further discussed. And the term peptide generally refers to a compound made out of 2 or more amino acids, up to 10, up to 50. And meanwhile, proteins, they're pretty similar to peptides. However, they're distinguished from polypeptides according to their size and their structure. They're generally containing 50 or more amino acids, and they're composed of several bonds, such as peptide bonds, disulfide bonds, and ionic bonds. So when peptides are only composed of peptide bonds, proteins are composed by more than peptide bonds. And you can see a simple figure here where the individuals or the small things are the amino acids, and then a chain of it makes a peptide, and a longer, more complex, and a secondary structure with different types of bonds are called proteins. Okay, and next I will be discussing how to name a peptide. This is pretty familiar. You just need to know a term called prefixes and also abbreviations. So the name of peptides are actually derived from the names of the amino acid residues. Peptides get their names from the amino acids, and the number of amino acid residues is often indicated by a set of prefixes up to 10. So we start from 2 to 10. On your right, you can see the prefixes and something called a terminus. So by convention, the name of peptides are always written from left to right, starting with the N terminus on the left to the C terminus on the right, where the last syllable of each amino acid name is replaced with "il," starting with the N terminus. Meanwhile, the C terminus retains its complete amino acid name. To make it easier, here's an example and also the abbreviations that has been previously previewed as well. So you can either use a 3-letter abbreviation and 1-letter abbreviation. And an example is a tripeptide consisting of alanine, glycine, and serine residues is named as, remember that a tripeptide, basically a peptide consisting of 3 amino acids, and alanine is soon named as alanyl, glycine as glycyl, and serine as it's located at the C terminus or at the right, it retains its serine name. And combined together, it's called alanyl glycyl serine, or using abbreviations, alaglycer. And this alanyl glycyl serine is all just one word. That's why most of the longest words in the dictionary are actually all proteins and all peptides, because you can combine all of the amino acids together and it still just forms one long word. So that's why most of the long words in the dictionary are proteins. Okay, and here's a simple way how to draw a peptide. So the first thing you need to do is to draw the structure starting from the N terminus or the left to the right, and then you can remove the O atoms from the carboxyl group and combine it with the right amino group, the H molecules, and form a water molecule, and then you link them together using a peptide bonds that will basically form your peptides. And for here, we see the structure of glycine, serine, and methionine. When you combine it, you have glycyl serine methionine. Okay, and here are several functions of peptides in our body. Peptides, we have a wide range of peptides that is applicable in the medical field and also already there in our body. These are a few functions of it. We have vasopressins that acts as an antidiuretic hormone responsible to regulate the amount of water in our cells by causing our kidneys to absorb the water. And we have defensins. This
as exactly like the name, defense. It's basically active in our immune system and promote wound healing. And then we have angiotensins that help regulate blood pressure, also hepcidin involved in controlling the body's iron absorption. Also, I'm sure you're familiar with some peptides like collagen that's used for our skincare to increase skin elasticity, and also oxytocin to that is familiar in the contraction of childbirth.
Okay, so this is now we're going to discuss about the formation of peptide bonds, which is, I believe, a continuation of the previous chapters. Sorry, of the previous subtopics. So basically, how is a peptide bond formed? Well, first, a peptide bond is formed by two amino acids that are covalently joined together through a substituted amide linkage, known as a peptide bond. And the peptide bond is this one, the one that has the CONH, the bolded one. And the reaction on the left shows that the alpha amino group with the R2 group, this one acts as the nucleophile, the one with the pink H. And then the amino displays the hydroxyl group in the amino with the R1 group. So that means the H will displace the OH so that the C and the N can connect. And it's also important to note that the carboxyl group must be chemically altered in some way to make it a more powerful nucleophile, as the hydroxyl, the OH group, as we know, is a bad leaving group. And basically, this continuation here, the one where where involves the release of water, it cannot just it not only happens once, but it can happen multiple times to form a long chain of peptide, which can be formed by multiple peptide bonds, as shown here. And each peptide, they have their own respective ends. The one with the positive end, sorry, it's called the amino terminal end, and the one with the negative is called the carbonyl terminal end. Next. And so these are just the properties of some of the peptide bonds, which are, the first one is that the carbonyl carbon is sp2 hybridized and has a trigonal planar geometry. And three, the amide bond has a certain structure known as a resonance structure, as shown in the figure on the bottom left. And because of the property of number 3, we have many derived properties due to number 3, which are two conformations on the peptide bonds, which are shown in the resonance structure. And all the atoms here, they lie on the same plane, and all bond angles are approximately 120 degrees, as shown on the picture on the right. And the NH and CO groups are oriented 180 degrees from each other. And this peptide bond, this double bond, can exist in as either cis or trans forms. Okay, next. And so these are the references that we use throughout the presentation. Next. And so that's all from us, Group C. Thank you for listening, and we would like to apologize for anything that we said or did that is of inconvenience to all of you. And so thank you and have a nice day.
Thank you. Yay!